That's not what shows with kind of positions with this society. Necessarily, to to the others for these series of decisions that you will see. So keep those because it's up to you to decide who's going to be our society in the future.
Tony, audio is not working.
That's interesting. Let me see if I can.
I thought it was just me.
Okay, I've tried. Does this work any better?
Much better.
Okay, we'll go with this microphone. Sorry, there's a couple of different microphones here. I guess some work better than others. Anyway, sorry, I'll just start. Not to belabor it, but I'm welcoming you, and also want to let you know that there's elections coming up. Keep an eye in your email inbox for the opportunity to self-nominate or nominate others. And then following a couple weeks after that, keep your eyes open for the opportunity to actually vote in the election. I also want to make the point, this is especially pertinent to our American members, but in case you haven't noticed, there is a major thrust by the NIH, among many other efforts, but probably the most pertinent effort is to increase the utilization of human models in research to the point where they are requiring the use of human models for the approval of NIH grants. I would just advise you to keep an eye on this. Keep your eye on the possible funding opportunities that emerge. But also, I think this is an opportunity for everyone who is on this call to try to make yourself the go-to person for human models at whatever center you are. And I think it could be a very useful time for us to integrate more functional precision medicine into a lot of the research that people are doing at cancer centers in particular. I now think we'll just proceed to introduce today's speaker. I'll ask you during this, if you want to do as I do and keep your camera on. I know I find when I'm in Jens's position that it's always a little bit easier when I see some faces looking back at me. So if you feel you are someone who can be observed, please put your, and if you want to, please put your camera on. So I'm very pleased to be able to present to you for today's lecture, Dr. Jens Kelm. He is the Chief Scientific Officer of PreComb Therapeutics, and he's also the co-founder of InSphero, a company that has pioneered microtissue-based assays and organ-on-chip solutions. He has received multiple national and international distinctions for his innovation and entrepreneurship, based in the development and application of 3D cell culture and organoid technologies for drug discovery as well as translational medicine. He has also been instrumental in establishing one of Europe's largest competence networks for tissue engineering and drug development, and serves on the steering committee of the International 3D Culture Conference organized by DECEMA, DECHEMA, which is actually where I met him at really an excellent meeting in Freiburg. I will say that for members of the SFPM, this organization is one to keep an eye on and one that I hope we can develop closer relationships to, because essentially the entire conference was about what we would call functional precision medicine. And there's a very, very significant overlap in the interests of this conference and this particular society. Dr. Jens' recent work is focused on the clinical implementation of organoid-based functional precision medicine with the goal of translating advanced in vitro technologies into tangible benefits for patients. So I will stop there, and I look forward. Dr. Kelm has told me that he is going to stop at intervals. He has a couple sections for his talk. So he is very happy to take talks at intermediate periods. And Jens, you just let us know when you see a good time for that.
Perfect, Tony. Thank you very much for introducing me and for this kind invitation to speak in this community. I think it's the SFPM initiative is a super society, and I'm really glad that we get the opportunity to show our concept, which we are pursuing forward, to be able to predict patient drug response. So the title of my talk is Dynamic Drug Response Profiling at Scale for Functional Precision Oncology. And I would say that the two most important aspects here is dynamic drug response and scalability, to ensure that we develop a technology platform which can scale at the end of the day into the clinic. And now we go. Good. As Tony said, I have different chapters in my presentation. I don't want to speak 45 minutes in one go in front of my computer screen here. So I will give you some intermediate breaks, or I give me some intermediate breaks, where you can address questions if there are any questions. I'm introducing the overall concept behind our screening method, which we have been followed over the past years. I would like to give you some case examples, how we are using technology so far, where we are standing in terms of validation of our assay platform. And at the end, I would like to give you a glimpse into the technological side, what we have done to optimize our test method, and how we would like to build a drug testing network based on our automation technology, not only here in Europe or here in Switzerland, in Germany and Austria, but also on a global scale. Good. I don't need to introduce functional precision oncology here, and that is super. That makes things significantly more easy for the introduction. So I was thinking, okay, how should I introduce our topic here in this community, and to make sure to present the different kind of perspective we have taken with our assay system. When we look at the technology platforms which are out there, most of them require statistical power to make predictions. There are platforms independent of which kind of cell model or tissue model is being used, looking into either dose response curves, measuring viability, or there are even platforms out there looking into single concentration treatments. Based on these dose response curves, a viability score is being generated, for example, the DSS score, which is used quite heavily. And then you need to define certain thresholds to define whether the drug sensitivity score is being supposed to be a responder or non-responder. To become predictive, you need a significant patient cohort to be tested upfront, to have solid boundaries and solid thresholds, which you can use to discriminate whether a certain score is being supposed a non-responder in the patient or a responder in the patient. The disadvantage here is that actually for every new drug or for every new combination you test, you need a new cohort to be tested to make sense out of the data. So what we were thinking of is, can we actually develop a platform which captures patient-specific drug response in a scalable manner without the need of having this statistical backbone a platform with which we can directly predict patient response in the same manner as if you would do in vivo animal experiments. And our design goals, our engineering design goals, were that we are flexible and efficient in the use of different cancer cell sources, that we can test a meaningful number of drugs per patient, and that we are capable of testing a high number of patient models in parallel. That's the scale, the scalability of the platform. As clinical relevance is being addressed by a number of different technology platforms out there, I would like to explain a bit what we here at PreComb understand when we talk about clinical relevance or capturing patient-specific drug response. We have designed the assay towards physiological relevance. So we are using the maximum concentration we are using is the physiological Cmax concentration of each individual drug which we are testing, if known. When we are testing exploratory drugs, they might not be known, but if they are known, the Cmax concentration is the highest concentration we are using to mimic more closely the plasma concentration in the patient. Drug response is not a single endpoint. Drug response is dynamic. And sometimes it takes quite a long time that you see a drug response in the patient. For example, for immune oncological drugs, the efficacy is not seen directly. It takes some time to unveil the drug response, or PARP inhibitors also take some time. So we are looking into a continuous and real-time monitoring of drug response dynamics. Based on that, based on the real-time monitoring of drug response dynamics, we can do an in vivo-like translational analysis, and we treat our data very similar as if you are doing animal studies, comparing treatment to control ratios and tumor inhibition growth curves and response kinetics. The incorporation of PK elements into the assay is also something very important to us, because it's not that the drug is being exposed in full concentration over 10 days or 14 days. Every drug has its individual clearance properties. At the moment, we can't do this for each individual drug we are testing. But what we are doing is we are dosing, we are removing the drugs, and then we are looking long-term how the drug response unfolds after the dosing of the individual drugs. And we are using 3D models of patient-derived solid tumors. So we are focusing mainly on solid tumors, and we are using three-dimensional models to test the individual drugs and drug combinations. And the reason is that efficacy is actually a result of both exposure and drug activity. So on the one hand, we have the biology, which is driving the drug response. But on the other hand, we have the pharmacokinetics, which is driving the drug response. Biology is being addressed very well in the past. We have 3D models integrated with all the heterogeneity of the tumor patient, with the genetic and the cellular heterogeneity. But the pharmacodynamic aspects, they have been treated only very marginally with most of the current platforms which are being used. And the pharmacokinetic aspects, like drug penetration into the tissue, exposure concentration, drug clearance, and dosing regimens, they are a major driver of the overall drug response. And I've been talking to the former global head of pharmacology at Roche a few months ago, and he even said that 50% of the overall drug response is driven by pharmacokinetics. So we need to have a certain elements of pharmacokinetics in the assay. So The basic principle is simple. We have our 3D model. We dose the drug in different concentrations, normalized to each individual Cmax concentration. We are measuring growth kinetics through imaging-based technologies just by bright field imaging over time frame of 2 weeks. And then we are looking into the individual growth kinetics or ideally shrinkage kinetics. And then we can do a response prediction based on that. So how does this look like? Just one example here. We have here on the top row, we have an untreated tumor tissue. And we distinguish between the response type, where we are analyzing the TC value, and the progression type, which is an adapted RECIST criteria. We have the untreated, which is heavily growing. We have drug A in the second row, which is effective. You see this, that even this tumor tissue starts to shrink, but then it relapses and we see growth over time. Then we have a drug B, and drug B is also super effective as compared to the untreated control, but it's only leading to stable disease condition. However, when we mix, when we combine drug A and drug B, then we see a significant accelerated and more effective response to the combination and leading to remission. And that's the therapies we would like to identify. Why is it so important to use the Cmax value of each individual drug? And I've plotted here the individual Cmax value of around, I think, 320 individual cancer drugs. And you see that there's quite a huge distribution in the concentration. We have cancer drugs which have a Cmax value of 0.0001 micromolar, two drugs which have 1,000 micromolar Cmax concentration. So we need to be very precise in using the individual concentration. 5-fluorouracil, for example, has a Cmax value of 450 micromolar. I don't know how the clinicians are getting so much drug into the patient, but that's huge. Other targeted therapies can be used in significantly lower amount of concentration. So the right use of the right concentration is a major determinant of the predictive capacity of our assay system. The way how we are analyzing our kinetic data is very close to the way how you are analyzing animal data. So the first step is we have our images, we have our image base, we are quantifying the images, we are calculating the individual microtumor volume, and then we get the image-based growth kinetics. Step 2 is that we are analyzing the treatment to control ratio to define the response type. And we discriminate between non-responder, weak responder, and responder. However, this does not tell us anything whether a drug is leading to remission or stable disease condition, because this heavily depends on the growth properties of the untreated control. So we have a second category, and that's the progression type. And that's step 3, where we are looking whether a drug is actually leading to a stable disease condition, to partial remission, or even strong remission. And we have set some quantitative boundaries here. So a non-responder is less than 10% difference to the untreated control is considered to be a non-responder. If we see a change of 10 to 50% as compared to the untreated control, it's a weak responder, and a strong responder is more than 50% as difference as compared to the untreated control. What is, I think, important here to understand that we are consider every screen as a kind of an individual clinical trial. So we have some certain quality measures in place that if the standard deviation of a single growth curve of a single drug is too high, we do not analyze. Untreated control is not stable. If we already see over time that the untreated control is getting smaller over time, then the whole assay is not valid and we cannot analyze the data. So for us, we have tried to build a statistical framework and a QC framework which allows us to draw actionable data from a single shot experiment. And just to give you an impression how this looks like in numbers, some of the audience might recognize this data. So these data have been generated together with Ina Ume at the KIDS in Heidelberg, where we have been testing a couple of pediatric cancer model systems. And this is part of the data we have generated together with Ina. It's an NTRK fusion pediatric neurofibrosarcoma model, and we have tested more than 50 drugs on this model system, each drug in its individual Cmax concentration. And it's an NTRK fusion, so the standard of care treatment is seletrectinib. It's an NTRK inhibitor. However, the NTRK inhibitor was a responder, however, only leading to stable disease condition. And the MEK-RAF inhibitor combination, in this case, tovorafenib and pimazetinib, was significantly more effective as compared to the standard of care treatment. And it was also significantly more effective to its monotherapeutic components, pimazetinib alone and also tovorafenib alone. One of the advantages I see using image-based analysis is that you can actually always correlate your quantitative measures to the images you have made. You can double check and see whether the quantitative data actually makes sense on the images or not. We have tested in the meantime more than 10,000 drug-to-patient conditions together with a number of our clinical partners. And this is just to give you a bit of an impression how the overall response segments look like, how the categories look like. When we are looking at the TC values, you see that only 20% of all of the drugs against all of the different model systems, adult models and pediatric models have been responded, have shown strong responsiveness, and 24% weak responsiveness. The most of them were not effective at all. However, when we look at the progression types, looks even more devastating because only a very minor treatments have been led to remission, and only 7% to complete remission, 4% strong remission, and 7% partial remission. However, these are the therapies we would like to identify for the patient at the end of the day. Good. This the method concept. Are there already any questions with respect to how we have set up the screening method, the test method?
If you have a question, please put up your hand. We have a few questions. Maybe Elad Katz, maybe you could go first.
Hello, I can see myself, but a great start, Jens. I'm very impressed, obviously, by all the work you've done. I have a lot of questions, but I'll stick to one because maybe you're going to answer some of them in the second part. You rightfully say that we should really look at remission versus responders, non-responders, which I think it's laudable as the right way to go. The question for me is really how you chose to have, if I understood it correctly, to have a single dose exposure and then follow up by 14 days, because that's nothing like what happens in the patient. So how did you get to that particular choice?
Yeah. So from the exposure time, we have looked at the different exposure times of chemotherapies and target therapies, and you are absolutely right. The dosing regimens of chemotherapies and target therapies, they are completely different. Chemotherapies are mostly given in intervals of 14 days, and then you cycle them. And also the clearance rate of most of the chemotherapies is significantly shorter than the clearance rate of most of the target therapies. Target therapies are giving every day over a 14-day time frame, or at least most of them. And the reason why we choose 14 days is a kind of a practical reason. We would like to circle back the data in a clinical relevant time frame. But it allows us for a more long-term dynamic observation of the overall response mix when we expose the individual tissues to the drug. So at the end of the day, we are absolutely aware that we will not be able to 100% reflect what is happening in the patient, but we try to find a good balance between patient relevance and practicality of the in vitro test and of the in vitro assay.
Okay. So just I'll let others to ask, but did I understand correctly? So for chemo, you do one dose, but for the targeted therapies, you're doing repeat doses?
Not, no. So we do single dose for targeted and for chemo, but if need, we can also do multiple dosings if there's a need to be. So what we have done is we have done 2 doses actually together with Ina, and the overall setup, and when I show you the automation platform which we have developed, so in principle it's possible that we can do multiple dosings. We can also sequentially dose two different drugs. So this is possible, but this is only possible because we have this longer period of time which allows us to play around with these kind of multi-dosings or sequential dosing of different drugs. But we haven't done this implemented routinely yet.
Okay, thank you.
Maybe we could hear from Dennis Plenker.
Sure. Thank you, Tony. Thank you, Jens, as well. I think like 2 questions, like a very short one. The data you've shown is right now on cell lines or on primary samples, and then the second is towards the AUC kind of, you said, like on your serum concentrations. It is super compelling for me kind of why you would use it, but don't you think that the exposure in a dish and even like on a patient by patient basis is different? And in the end, I feel the data is going to show, right, like what's right or wrong. But some chemotherapies I think have like millimolar levels kind of floating around in the body, and they still kind of, you still have resistant patients, right? But well, you would just get toxin culture. So just curious to hear your thoughts on that.
Yeah, no, you are absolutely right. That it is an approximation to the patient concentrations. However, we need to start somewhere, right? We do work together with the university hospital in Aachen to further refine our exposure concentration and exposure time for individual drugs. However, if you would like to test 30 drugs in parallel on a multi-well plate, you need to make certain compromises. And we are absolutely aware that it's not perfect yet, but it's a step towards the better representation of pharmacokinetics in a high throughput assay. Your first question, I think I will answer this in the next slides. So the data I've shown is from patient-derived cell lines. Actually, they have been originating from Ina's repository of cell lines. But we have done both, and that's the flexibility of the overall assay platform, is we can work with either standard cell line spheroids with patient-derived cell lines, or with tumor models derived directly from fresh patient tissue.
Okay, cool. Thank you.
Great. Finally, Mike PreComb.
Thank you. Yeah, one quick question. Just how frequently do you see the combo be more effective than the monotherapies? That data just caught my eye.
And this strongly depends on the combination. For some combination, we see this evolving very quickly. And for other combinations, it takes some longer time when we see the combinatorial effect. What we actually have seen is that whether there is a synergy or whether there's not a synergy, it's super patient specific. It's not that that two drugs are per se synergistic. What we see is that the MEK and RAF inhibitor, I think we had only 1 or 2 patient models where we see synergistic effect of those drugs. But what we also see is we see additive effects, right? At the end of the day, the patient doesn't care too much whether it's synergistic of additive or additive. If the combination is more effective than the monotherapies, I think that would be the way to go. But yeah, it depends on the combination at the end of the day, how fast and how quickly we see whether they are having a beneficial or an additive / synergistic effect as compared to their monotherapeutic components.
Great. Yeah, I'm going to have some follow-up questions, but I'll hold off till later.
So if the time is not enough for today, because it's already past, I see, and I think 1/4 through my presentation, then we can discuss this also offline. So if there are any pressing questions or other.
I think you can continue, Jens. I'll just ask the audience. Everyone check real quick. Make sure you're muted if you are not, and turn your mute on if you're not muted. Please go ahead, Jens.
Okay, so I will accelerate here a bit my speaking speed. I think that plays directly into Dennis' question, whether we can, what's the source material of our model systems? And the answer is we work with both. And I think it's important that our platform can work with both kinds of cell sources. Ideally, you get enough tissue that you can produce your model system directly from the fresh tumor sample or from the fresh biopsy to mimic not only the cancer cells, but to mimic the overall individual tumor microenvironment of an individual patient. Each cancer is differently composed, and this different cellular compositions needs to be reflected ideally in an ideal case. But there are tumors, and there are where you don't get enough cancer cells from. We work with a DIPG cancer and pediatric DIPG cancer here in Zurich, and they get only minor amounts of cancer tissue from the patients, and they need to expand the cancer cells to be able to test drugs. And we can use both. We are working for both in a 384 well plate format, and this allows us to flexibly design the drug layout which we would like to test. We are testing up to 32 drugs in 3 different concentrations on one 384 well plate. These are a few examples of microtumors which we have produced from fresh tissue: glioblastoma, colorectal, liver, pancreatic, PDX, pancreatic resection, DIPG, biopsy. So I think from the model manufacturing perspective, we have sufficient leverage that we can work with a broad band of different solid tumors. When we are working with fresh tumor tissue, then we can maintain all the individual cellular components of the original tissue. And this is a work which is currently in preparation for a publication. What we have done here, we have used non-small cell lung cancer, digested it enzymatically, produced the tumorid or microtumor, and then we perfect over 6-day-old microtumors. And you see here on the right side the results where we see the lymphocytes, the epithelial cancer cells, NK cells, CD8 T cells, macrophages, B cells. We lose the B cells over time though, and also connective tissue cells. And we have done this now for, I think, 12 or 13 NSCLC patients. And for all the patients, we have been quite successfully maintaining the immune competence of the tissues over time, so that we can not only test chemotherapies and target therapies, but that we can also test immune oncological therapies. I move a bit quicker, otherwise I will not be finishing my talk in the next 1 hour. So we have, together with our clinical partners, and a number of them are here listening in. So by means of Ina from the kids in Heidelberg, Jasmina from the NCT in Dresden. I've seen Heike. I've also seen collaboration partners here from Zurich with Sylvia. And with all those collaboration partners, we have looked at different aspects of the validation path. One of them was that we have been using, together with Ina, reference models with known genetic alterations. And the hypothesis is that when we have a certain genetic alteration, the corresponding targeted therapy should lead to a response. And this is shown here for the same NTRK fusion myofibroblastoma model, where we have been using an NTRK inhibitor. In this case, we have been using the entrectinib and not zelicapavir. And you see clearly there's a clear response of the targeted therapy of entrectinib. Strong remission, a strong responder, however, progression type stable. And we have done this for a couple of more individual model systems provided by Ina and her team. And we have looked at the different responses at 1 times Cmax, 0.1 times Cmax, and 0.01 times Cmax. Compare that to non-cancer reference model. That's a connective tissue model based on the cell line VH7. And we've been looking whether we see any unwanted toxicity of these treatments. Overall, summarizing, we have had 14 reference tumor models with 14 unique genetic alterations. And in total, 26 drug target correlations. We had, in most of the cases, more than one target therapy targeting the same target. And overall, we have seen a sensitivity of 85%, and looking at the non-tumorous tissue screens, a specificity of 95%. And I think that looks quite promising. We have also started to look into direct translational studies. And this is one data deck which we have generated together with our partner here at the University Hospital in Zurich, the pediatric oncology with Professor Dr. Javad Nazarian. And they have established cell lines from pediatric brain cancer, from DIPG cancer. And I assume that most of you are familiar with pediatric brain cancer. This is a very severe cancer, and there is no treatment. And Ina, you can interrupt me and correct me, but it's ultimately the outcome is fatal. We had the patient history of this patient, and we have done retrospective correlation, whether we see the same kind of response in our assay as compared to the patient. And the patient underwent temozolomide monotherapy, temozolomide, lomustine combination, and a triple combination, including osimertinib. None of them were effective at all. Looking at our results from our screen platform, we also don't see any impact of these treatments on the growth of the tissues. Then this patient was enrolled in a clinical trial, and it received an experimental therapy, ONC201, which is just being approved for pediatric brain cancer. And in our assay, it was super sensitive towards ONC201. It was super effective. And the clinical outcome of the patient was 19-month stable disease conditions. And I assume that Ina will confirm that, but 19-month stable disease condition for a DIPG brain cancer is significant. It's really significant. Then we have been able to get actually tissues from the same patient after the patient has become resistant against this targeted therapy. And we have been producing tissues directly from the fresh tissue this time. And we see progression in the control. And also, unfortunately, the target therapy was not effective anymore, which was highly effective before the patient has acquired drug resistance. We have been talking already about combination treatments, and we might ask how long does it take to see whether a therapy is being more effective in the combination as in their single counterparts. And this is one example here, actually, where the difference start to show up after 5 to 6 days, where we really see a difference. And in this case, a clear synergistic effect of two therapies which are not effective on its own. And we can analyze this over a couple of different model systems. This is over different brain models, pediatric brain models and adult brain models. And you can get a bit of a broader understanding whether a combination is being more effective and whether the combination is leading to more remission outcomes as compared to the individual, in this case, RAF and MEK inhibitors. Another case example, as I already presented, when we are using fresh tissue, we maintain the immune competence of the original tumor. So this allows us to test immune oncological drugs. And one example is here, two different immune inhibitors, Keytruda and Tecentriq, PD-1 and PD-L1, exemplified for 2 patients with a high TPS score and a low TPS score. The TPS score is being used in the clinic to assess how likely a checkpoint inhibitor is being effective in this patient. The higher the TPS score, the higher the likelihood that the patient is being responsive. And the TPS score of 40% is good. And we also see here, for the non-small cell lung cancer patient, we have tested here that both Tecentriq and Keytruda have been highly effective. They have been similarly effective as an approved chemo combination, which we have been using as a control. Exemplified for the second patient has a lower TPS score, and there we see, yes, there is effectiveness of the checkpoint immune inhibitors, but by far not as being effective as the chemo combination therapy. One of the NSCLC patients, which we have tested together with the Medical University in Innsbruck, actually got a relapse and got back into the clinic and were then treated with one of the triple combinations we have been tested on his tissue before. And the clinical outcome of this treatment, of this triple immune oncology treatment with pembrolizumab and chemotherapies, was stable disease partial remission. And you see here the blue line, this is the triple combination outcome which we have seen in our assay, which has also led to stable disease / partial remission outcome. So we are accumulating at the moment anecdotal translational case examples that there is a high probability and a good chance that we are capable of predicting the patient outcome quite precisely. However, these are not clinical trials, and we are absolutely aware of that. And this is something which is in development. One of the last cases that I would like to show on to be advised here at the University Hospital in Zurich. We see here the aqua control, the growing tissue, and then we have added different concentrations of CAR T cells, and you see here the dose-dependent effect of the CAR Ts. The CAR Ts proliferate. That's why it looks like the tissue is growing, but these are the proliferating CAR T cells in the system. We have done as a control untargeted, unspecific T cells, and there we do not see the same kind of proliferation effect. And then we have been evaluated the death rate, the viability of both the unspecific T cell control and the CAR T, and we see a very nice dose-response correlation. The more CAR Ts we used to a certain stage, the higher the death rate of the cancer cells have been, which was not the case for the unspecific T cell control. Where do we would like to go from now? As I think we have made quite good progress on showing the potential to identify the right therapy for a patient. However, we think that an in vitro assay cannot only inform on the right therapy, but can also inform on the dosing for the patient. And this is something we are currently looking at together with Professor Dr. Lars Küpfer in Aachen, whether we can use an individualized PBPK model to inform the experimental outline by means of dosing and timing of the in vitro assay, and then derive dosing information for the individual patient. This is early stage. We have a proof of concept for docetaxel, but this is something which we are pursuing in the next weeks and month. And before we are getting to the automation, are there any questions with respect to the case examples I've just shown?
If you have a question, please put your hand up. Otherwise, we can let Jens continue. Dennis.
Sorry, it's me again. I have another question now, just because you said now you have kind of the patient models. Are you still then following all the growth just via bright field imaging, or do you now add a lot more markers? Because in the end, you just want to focus on your tumor cells, right? So in the end, you run into the problem of who's a tumor cell, who's not.
Correct. So the core principle is that only tumor cells are proliferating in the system significantly. And this is something I've seen in the past, that stroma cells do not proliferate. So it's mainly, which is contributing to the increase in size, it's the cancer cells, it's not the stroma cells. So no, we don't use specific markers because we would like to still have a scalability in the system. But we do not destroy the tissues, monitoring the growth over time. So what you can do is you can add any kind of different downstream analysis. So what we do, for example, we have established here single tissue RNA isolation. So you can look into transcriptomics, or you can add ATP assay to confirm your growth kinetics. You can even do a high content analysis if this is something you're interested in from the research perspective. But from the clinical implementation perspective, you would like to keep it as simple as possible.
Sounds good. Cool.
Sean Mahoney has a question.
First of all, fantastic presentation. This is incredible work. The question that I have is this, or could this testing be available to anybody having surgery anywhere in the world, notably pediatrics? I'm not an MD, I'm not a PhD, I'm on the patient-parent side of things, bereaved, unfortunately. But I've worked with close to 200 ependymoma families and other types as well, where we're just trying to find a way to, test not only the single drugs, chemotherapies, but also combinations and immunotherapies. The immunotherapy piece was fascinating to me.
That's a super question, Sean, and it leads actually to the automation and to our overall basic concept. Because one of the, I think, on the core of the core strengths of functional precision oncology in the past was that we had only centralized facility who were capable to perform the test. We would like to enable clinical institutions to do it by themselves. We would like to have it decentralized. And this would have been my call for action at the end of the presentation, because everybody who would like to work with us, I would like to invite to come and to talk with us, to become part of this decentralized drug testing network we would like to build. And core of it is that we do have automation which fits into even non-expert users, I would say, and I know you're all scientists and you're all experts, but probably not automation experts. So what we have done is we have developed an automated an automation device where you just need to load your test kit with your drugs and your medium, and you have to load your plate with the tissues. Then you give it a name, and you scan the barcodes so that the machine knows which test kit belongs to which tissue plate, and then you give it a go. And after 2 weeks, you come back and you pick the tissue plate out of the device, and then you can do any other additional test you would like to do. The device you see here, it looks like a tank, and it's not very pretty, but it's powerful. And it's powerful because we can load whenever a tissue sample is ready without interfering with existing screening processes. And we can run up to 40 patient plates in parallel. That sounds trivial, but we have been talking to a number of liquid handling providers, and this sequential discontinued or continuous loading of samples is not trivial from the software control software. The analysis is done fully automated in the cloud, and we are currently working with AWS and with Reactor to provide access through the cloud to the individual for our individual partners and clients, that they have full access to the data, and at the end of the day, to the data bank, which is being created from the data. I skip that in as we are running really short on time. So this is what we currently building with AWS and Reactor, being a cloud-based accessible system. And this decentralized model, I think, brings us a huge opportunity to work towards more quantitative precision oncology, which I think is significantly important in developing new drugs and to integrate real-world in vitro drug testing early in the drug discovery process, and to accelerate and to de-risk the overall drug development pipeline. And I know we have people here from Roche who can comment on that as well. And this is what I mentioned. What we are now trying to do is to use our automation more transforming clinical science with cell banks into testing hubs and working together with them, not only for clinical applications, but also for using this together with pharma companies and allowing them to screen their drugs quickly on a vast variety of diverse patients. And we do this not alone. To build this ecosystem as a small company, this is hardly doable. So we have been partnered with TCAN, with Reactor and AWS to execute that. And yeah, so this is my call for action, and this is actually also the end of my talk. that we are seeking for clinical institutions who are interested to work with us and to discuss with us to initiate a global network for dynamic longitudinal drug testing. And with that, the time went on much more quickly than I thought, so it was super engaging. I would like to acknowledge all of our clinical partners. Many of them are in this call here. So personal thanks to Ina and Yasmina, to Silvia for their ongoing support, and for the great collaborations we have.
Great. Thank you, Jens. That was really excellent. I love the way you're actually addressing the issue of trying to decentralize this. I know that for some of us, myself included, I find it challenging enough to do it in a centralized fashion, and to go a step beyond that is going to be important ultimately. We have time for maybe one question. Are there any questions from the audience? I see a hand raised by Elad Katz. Maybe you could ask.
Hello again. Excellent. And I agree. I have a question that is, I guess, linked to your call. So this profiler machine that you have, is your vision that it's going to sit in every major cancer hospital? Does it require, again, I'm going back to my original question, is it a matter of you plate the cells, you put one dose on, then you just follow it? Or because in 3D, everyone that had hands-on experience would tell you it's all about the media changes, right? Do you do any of that, or is it really just something that in a hospital setting one could plate and forget about?
At the end of the day, it should be exactly like you said. You plate the cells, you put it into the profiler, and the profiler is doing medium exchanges. We can't do a 2-week test without medium exchange. So the automated system is doing the dosing, removal, medium exchange, imaging, incubation. We have a storage for the drug plates, for the medium plates incorporated into the system. So it's a fully automated screening system. What we now are going to integrate is the automated manufacturing of the tissues from fresh tissue material. This is still missing, but this is something we have started to think about, how we can also simplify the production process towards a robust production of microtumors, which are reflective of the original native tumor of the patient.
And do you think that this is the step where you would be able to put this in various locations?
I think it's already possible to put this into locations. There are so many clinical hospitals, like the kids in Heidelberg with Ina, like the NCT in Dresden. They are already experts, so there are already a number of university hospitals where we can place already the technology now.
And I think with that, we need to wrap things up. Thank you, Jens. I would ask every single person, if you enjoyed today's content, or actually even if you didn't, but I don't see why you wouldn't, please check to make sure that you are an SFPM member. Go to the site, sfpm.io, log in, and please join. We need to keep our membership as high as possible. So thank you, everyone, but especially thank you, Jens, for a really outstanding talk. And we will see you all next month.
Excellent. Was a pleasure. Thanks for inviting. Cheers.
Bye. Thank you.